My name is Olivia Moeck, and today we're going to be talking about adaptive immunity, but more specifically, the role of B cells and antibodies. Let's get started. A B cell is sensitized by exposure to antigens. Let's first look at the structure. So these red dots represent antigens, the blue are antibodies, and the green is class 2 MCH. Once antigens are bound to the antibodies in the B cell membrane, the B cell displays those antigens into its plasma membrane. Activated helper T cells encountering the antigens release cytokines and can stimulate the sensitized B cell and trigger its activation, seen here. It's important to remember that this is a class 2 MCH, and when it encounters it into a T cell receptor. The activated B cell then divides, producing a memory B cells and plasma cells that secrete antibodies. Now, let's look further into the antibodies. We'll now be focusing on the actions of antibodies. Number 1 is neutralization. Both viruses and bacterial toxins have specific sites that must bind to target regions on body and cells before they can enter or injure those cells. Antibodies may bind to those sites, making the virus or toxin incapable of attaching itself to a cell. This mechanism is known as neutralization. 2 is precipitation. When the antigen is a soluble molecule, such as a toxin, this process may create a complex that is too large to remain in solution. Put simply, the formation of insoluble immune system complexes is called precipitation. Is complement. When the antibody molecule binds to an antigen, portions of the antibody molecules change shape. This change exposes areas that bind complement proteins. The bound complement molecules then activate the complement system, which destroys the antigen. Number 4 is phagocytosis. Antigens covered with antibodies attract eosinophils, neutrophils, and macrophages. These cells phagocytize pathogens and destroy foreign abnormal plasma membranes. Number 5 is opsonization. A coating of antibodies and complement proteins increase the effectiveness of phagocytosis. This effect is called opsonization. Number 6 is inflammation. Antibodies may promote inflammation by stimulating basophils and mast cells. Number 7 is adhesion. Antibodies dissolve the saliva, mucus, and perspiration coat epithelia, adding an additional layer of defense. A covering antibodies make it difficult for pathogens to attach and to penetrate body surfaces. Lastly, we'll be defining haptens, epitopes, and complete antigen. Haptens are non-antigenic molecules, low molecular weight, that cannot induce an immune response alone, but react with products of that response. Epitopes, however, are a site within or on an antigen that stimulate immune response. Response is also direct. Then we have our complete antigen, which induces a humoral or cell-mediated response, immunogenicity, and interacts with the products of the response. That's all we have for you with adaptive immunity, specifically with antibodies and B cells.